Hey, welcome back. Mr. P here. Chapter 7, Lecture 3 is the third lecture in the Chapter 7 series. In the first lecture, we talked a little bit about the cell theory and the people that were instrumental in the development of the cell theory. In Lecture 2, we talked about cell surface size and surface area to volume ratio. Now we're going to talk about prokaryotic versus eukaryotic cells, which is one of the fundamental or foundational type pieces of information that cytology is based on. And my friend Mr. Dino here is kind of confused, and so you might need to help him out. When asked about what the opposite of prokaryote is, he thinks it's conkaryote because the pros and cons are opposites. However, prokaryotic versus eukaryotic, prokaryotes and eukaryotes are the opposite types. Okay, prokaryote is a cell that is very ancient in cellular structure. A eukaryote is a much more modern cell, as we'll see in a minute. Okay, so a little bit about prokaryotes. Prokaryotes at a base word definition level, pro is a prefix that means before. Eu is a prefix that means true, and karyon is Greek for kernel, and it stands for nucleus. And so when you start putting these together, prokaryote is a cell, or is a name for a cell, that is structured like a cell would be structured prior to the development of a nucleus, or prior to the evolution of a nucleus. Eukaryotes would be the post-nuclei version of the cell. And so U is true, karyon is kernel, and so the eukaryotes are true nucleated or true nucleus cells. And so when we talk a little bit about how these words kind of give us hint into when in history these cells would be seen, prokaryotes would be seen a long time ago. Eukaryotes would be seen more recently in history. Okay, and that's really important. Prokaryotes before the nucleus, eukaryote is after the nucleus. Hey, mister, just because you are a eukaryote does not mean whatever you say is true. Okay, that was my little bacteria mad sound. Obviously, when we look at a microscope here in a couple days in class, they're not going to be yelling at you, and you can pretty much well do whatever you'd like to them, as long as it's ethical and as long as you are staying within the parameters of the lab. Okay, prokaryotic cells. So what is a prokaryotic cell, and what are the structures that are indicative of prokaryotic cells? One, like we've already established, is that prokaryote is the ancient structure of the cell. And so there is no nucleus. There are no membrane-bound organelles. It's a very ancient type structure, and is very basic and simple. Okay, prokaryotes are on average 2 to 8 micrometers in diameter. Micrometers. Those are very, very small. You obviously need a microscope. Light microscopes work really well at distinguishing between prokaryotic cells. Okay, we can use light microscopes in order to determine relative sizes and ultimately relative shapes of prokaryotic cells. In a prokaryotic cell, the DNA is coiled into a region that we call the nucleoid region. Now, why is a nucleoid region not considered a nucleus? And it's simple. There isn't a membrane. And a nucleus, by definition, requires a membrane. Okay? And if I can get my pen to work. I'm in Trebs. My computer's freezing up on me. Stay patient. Okay, pen. Here we go. So, DNA is coiled into a region that we call the nucleoid region. It is because there is no membrane around it. And if we don't have a membrane, we cannot call it a nucleus. Okay? Only call it a nucleus if it is enclosed with a membrane. And we'll find out in a little bit when we talk about eukaryotic cells that membranes around the nucleus are actually double nucleated. Okay? It is a really thick membrane with pores, so it allows things to go in and out, but you ultimately want to keep the DNA safe. Prokaryotes don't keep their DNA as safe as eukaryotes, and it leads to a lot of genetic problems, or can, but they combat that with fast doubling times. They combat it with large sample sizes, And that'll all make sense, or more sense, when we get into evolution and natural selection later in the year. Okay, so prokaryotic cells, within their membrane or within their cell wall, they would have cytoplasm. That is indicative of all cells. Okay, cytoplasm is the jelly-like fluid that fills up a cell that all the membrane-bound organelles, or all the non-membrane-bound organelles, and all the DNA is kind of bathed in. It's kind of like Jello, or a really thick kind of jelly, and it gives structure and it gives support to the cell. Plasma membrane surrounds the cell, and not only the plasma membrane, but in most cases a cell wall is present outside of that cell membrane. So in a bacteria cell, and I have a picture you're going to see in a minute, but on the outside of a cell membrane, around the outside of a prokaryotic cell, you're going to have a cell wall. Now these cell walls are very much like the cell wall in a plant cell, but they're composed of different materials. A cell wall in a plant cell is composed of cellulose. We talked about that in chapter 2. Okay, cellulose is a polysaccharide. It's a structural polysaccharide used in cell wall construction. But in prokaryotic cells, the cell wall is not composed of cellulose. It's actually composed of peptidoglycan. And we'll talk a little bit more of that later. On the outside of a prokaryotic cell, there are flagella and cilia, and both of those aid in movement, and they both have different structures. So if this is a cell, if you see a long single projection, or you see a group of long projections coming from typically one location on the cell, we call these flagella. Okay, that is a flagella. You can see cells that have multiple, you can have cells that have one. But the flagella is a whip-like propeller. It's a whip-like tail, and it allows those cells to move through their environment. And one of the things that you typically associate these types of structures to is a sperm cell. Those cells would not be able to move without the whip-like motion of that tail. If you've ever seen a snake in the water, or a snake in the grass or on the ground, it's the same kind of thing. It moves back and forth, side to side, propelling the cell through its medium or through its environment. Cilia is a different structure. Cilia is a cell, but you see all these tiny little small attachments, okay, or structures, and they typically are all the way around the cell, and these move materials around the cell. So these flagella are in charge of moving the cell around. Okay, and put an arrow here. These cilia aren't as associated with cell movement, but in moving materials around the cell. Okay, so if we think about maybe a square here, and this is a food molecule, and this bacteria cell wants to get this food molecule around the side of the cell so that it can enter in through a pore here, or in through a channel protein, or in through an oral groove, or whatever we're talking about. These cilia move just like the wave in a football or soccer stadium. Okay, if you can imagine that. If everybody's waving in one motion, it kind of travels around the stadium in kind of a rhythmic motion. Well, these cells have the same types of microstructures, and will move materials around the cell and enter the cell in a specific spot. Okay, that is the job of the cilia. The job of the flagella is to move the cell through its environment and medium. Pili is another type of structure that's attached to the outside of a cell membrane and cell wall, and it aids in the conjugation process, which is a kind of a transfer of bacterial genetic information, or the bacterial plasmid, during asexual reproduction, and that's always in the form of binary fission. You don't need to know that right now. We will talk about binary fission, asexual reproduction, sexual reproduction in chapter 10, which is towards the end of this semester. Okay. Here is a picture of a prokaryotic cell, and all of the structures that I just talked about are in this diagram. Notice that these structures right here are the flagella. This one has 3. These would be pili, and can also be cilia, right? Pili are typically used to attach it to the surface or attach it to its environment, and during conjugation can move information from one cell to another. There's always a plasma membrane. Okay, that's a cell membrane. It's this little kind of blue interior membrane right here. There are cell wall, and this thick yellow structure on the outside of the membrane is the cell wall, and it's composed of peptidoglycan. There are ribosomes, which are not endomembrane organelles, but they're really the only organelle bacteria have. So that's going to be a key idea for you to know. Okay, ribosomes are bacteria protein makers, and ribosomes are eukaryotic protein makers as well, but they're going to just they're going to work with the mRNA, they're going to work with tRNA, they're going to work with the transcribed RNA messages and produce protein, but that again is going to be something in later chapters. For now, know that a bacteria cell has a nucleoid region. Again, no nucleus because there is no membrane around it. The nucleoid region is a densely packed DNA ball, and this is a chromosome that is kind of wrapped up, kind of like a wadded up ball of string. And these ribosomes, which are 70S, also important. Bacteria ribosomes are 70S ribosomes. On the inside of this cell membrane, you have the nucleoid region, ribosomes. This little tan colored is the cytoplasm. The cytoplasm is that jelly-like fluid. On the outside of the cytoplasm is the cell membrane. On the outside of the cell membrane is a cell wall composed of peptidoglycan. On the outside of the cell wall are the cilia or pili, or in this case, flagella. And all of these things play a really important role in the structure of this cell and allow it to do the things that bacteria cells can do. So, a little bit about prokaryotic shape. There are two main shapes that we want to know about in this class. One of them is a cocci bacteria, and cocci is a spherical shape. It can be either Staphylococcus or it can be Streptococcus. Now, Staphylococcus is a cluster of spherical DNA or spherical prokaryotes. Streptococcus is a chain of spherical bacteria cells. So Streptococcus, chain, Staphylococcus, a cluster. And so some of the things that you might remember or recognize is Staphylococcus aureus is a strain of bacteria that causes staph infections, and you might be able to look up MRSA or other strains of bacteria skin infections. Those are these types of bacteria. They are clusters of spherical bacteria. These are Streptococcus, and Streptococcus would be things like strep throat. Strep throat is a disorder or disease that, or an illness, I should say, that is basically, Streptococcus is caused by bacteria that are shaped just like this. Some of the bacilli, these are sticks or rod-shaped bacteria, and when you look at a microscope, you're either going to see bacteria that are circular or you're going to see bacteria that are rod-shaped. And the ones that are rod-shaped can either be Streptobacillus. Okay, again, strepto meaning chains, and if it's Streptococci, it'll be that. If it's Streptobacilli, it'll be this. Both of those are chains. Staphylococcus would be that. Streptobacilli would be that. Again, just know cocci and bacilli. Bacilli are rod-shaped, cocci are spherical. Little joke for you. How do you move an organism from one end of the room to the other? You carry it. You get it. Okay. So, eukaryotic cells. Isn't that clever? Eukaryote. Not prokaryote. Eukaryote. Like, if you want to carry that, eukaryote. Okay. So, eukaryotic cells, all other cells, basically are things like plant cells and protist cells and animal cells. All of those things belong to the eukaryotic cell class or the eukaryotic cell kingdom. And eukaryotic cells are the cells that make up you, so they're probably the ones that are most familiar to you. all of the cells that are in eukarya have membrane-bound organelles, okay? And the cells themselves have a cell membrane. It's a pretty standard structure that all cells have a membrane, because what is going to keep the cytoplasm in if they don't have a membrane? Again, if they have a membrane, they're going to be filled with fluid, and that fluid that they're filled in or filled with is cytoplasm. Again, that's the jelly-like fluid that kind of houses and supports all the organelles. Structures within the cytoplasm are organelles. This is a key distinguishing difference between prokaryotes and eukaryotes. Organelles, specifically endomembrane organelles, are in and always in eukaryotic cells and never in prokaryotic cells. Organelles are places and compartments that has its own specific function, specifically its own job. So the Golgi apparatus is an endomembrane organelle that's in charge of protein modification and protein packaging. The rough ER is in charge of working with the ribosomes to actually produce the proteins. The smooth ER is in charge of producing lipids. And so you can see in all of these cases there are specific organelles that have its own specific job. Again, structure dictates function. You hear that over and over and over in this class, and it's true with organelles as well. Eukaryotes can live alone as unicellular organisms or multicellular organisms. If we look at those prefixes, unicellular means one, multicellular means many, and so this picture right here is an example of a paramecium, and a paramecium is one single cell. It lives just like you, just like me, just like a cat. It has all 8 of those characteristics of life, and it's composed of one cell. Okay, that's a unicellular or single cellular organism. Multicellular organisms would be things like us, plants, animals, fungi, and protists. Okay, single-celled organisms, we just said, are protists. Those are things like euglena and paramecium and amoeba. Multicellular organisms would be things like plants, animals, and fungus. And so if we think about all the multicellular types of plants, or the multicellular types of animals, or the multicellular types of fungus, I leave you with one question. Which eukaryotic organisms throws the best parties? The answer is always the fungi. Eukaryotic cells will look like this. Now, I'm not going to go through all the organelles, and I'm not going to go through all the structures of the organelles and or the specific functions of the organelles, but I want you to recognize this cell and how it's a lot different and more complex than that first picture I showed you. Notice there is a nucleus. Notice you have all these different organelles, and they're all jam-packed in here. It's very much more complex. It's more modern. It has a greater size. And in general, just a more robust cell. Here's a plant cell. It would be virtually similar or virtually the same as a plant or an animal cell. The only difference is it will have this thick cell wall around it. Remember, the cell wall is composed of cellulose. And another big difference would be these green things. If we go back here, there are no cell walls around this cell, and there are no green organelles. These green organelles are specifically the chloroplasts, and those are going to be the organelles that are in charge of photosynthesis. Makes sense. Animal cells don't have photosynthesizing organelles. Plant cells do. So, getting into a little bit about the specialization and differentiation. Another joke for you. Stem cell parental advice. You can be anything you want to be when you grow up. And if you know anything about stem cells, is that they are undifferentiated and unspecialized, meaning that stem cells can become anything you want them to be. That'll do it for this one. Mr. P out.